Hi, everybody. Thanks for the introduction. I'm Alessio Crippa. I'm working as a postdoc at Karolinska Institutet in Stockholm. And today I'm going to talk about the implementation and the analysis design of an adaptive outcome trial in R. So I have many points that I would like to cover, and I know that I don't have enough time to do that. So I will leave the link for the slides and at the end as well, if you are interested in getting in more details on the topic. And today I'm going to talk about how to design and analyze a platform trial, and I will present as a working example a real platform trial that I'm working with. So the general idea is, why do we need a platform trial and what it is? Why can't we implement a traditional clinical trial that is actually the gold standard, where we are interested in evaluating the one therapy for one specific disease? And this is usually the gold standard. The problem is that most of the time we are not interested in just one treatment, and we are also interested in addressing multiple questions. And here where it comes many limitations using a traditional clinical trial. And this is particularly true when you have a heterogeneous population and several treatments to be compared. And as a result, usually you have very high cost for running this clinical trial, and there is also like a slow progress time, and usually you have a very high failure rate, also at the later stage of the development of a drug. So a platform trial may help to overcome those limitations. And usually a platform trial is a skeleton, so it's like a structure to address a specific disease in a very heterogeneous population where multiple treatments are available. And here I put a table from a Berry paper contrasting the idea of a platform trial versus a traditional clinical trial. So usually you have a specific disease, and you want to evaluate the efficacy of multiple treatment against just one treatment, and it's potentially long-term because many more treatment may enter the trial, as well as all treatment may leave the platform because they are either have a lot of evidence in favor of the effect, or because, on the contrast, can be removed for futility. And generally, also the randomization is implemented as a responsive adaptive, and I will talk more about that later on. So what I'm working with is the ProBio, and it's an outcome adaptive randomized. So we have multiple treatment, biomarker-driven platform for patients with advanced prostate cancer. So the background idea is that patients at a later stage of prostate cancer may benefit from multiple treatments that they have been already approved for their cure. But the problem is that they have shown to have very benefit for the patient population, meaning prolonging the progression-free survival times. But on the other hand, their effect is very heterogeneous. So some patients benefit a lot, while other patients, they do not benefit at all. And in addition, these treatments are very expensive. So the idea is like, looking at retrospective studies, we see that if you look at the tumor properties, so at the biology or the profile of the tumor, you see that people with a specific alteration, they may have a longer survival. So the idea of this trial is, can we use this information also to choose which treatment to give you, and also which sequence of different treatment to give to the patient. So this is the main objective. So try to use this information to decide which treatment to give you, and to see if this action will be able to prolong the progression-free survival time of the cancer. And then a bunch of other secondary aims. And I didn't stress enough, this is like a multi-center platform trial covering many hospitals in Sweden and also in the Nordic countries, so expanding to Belgium and hopefully to UK. And this is the general framework of the platform study. So there is patient coming, and then these patients are randomized either to the control group, that is meaning like give the patient to the doctor without saying what is the genetic profile of the tumor, while other patients are randomized to one of the treatment depending on the genetic profile of the tumor, and then to constantly update the outcome data and use this information to evaluate the therapies within the signature, and we'll talk soon about that, what I mean with that, and decide if I need more patients because I didn't see enough data, or if I have seen enough and I can stop the randomization to that treatment in that signature because the treatment is very effective or the contrary is not effective at all. And then while treatment graduates, meaning that is beneficial then to introduce a validation trial or to add new therapies and throughout the time. And what I do while I evaluate the treatment is also to update the randomization probabilities in order that a new patient will benefit from the outcome of other patients and will get more likely a treatment that is more beneficial for him. So this is one of the most complicated slides that I have here. So we have 4 types of binary biomarker group in this table, and you see that a patient may have multiple mutations. So a patient will end up in just one of these rows when it gets randomized. The problem is that if I want to evaluate the treatment at this level, then I don't have enough power because some of these combinations are very low. So what I'm actually interested in is generate the idea of the main effect. It's not really true, so it's just like a combination of these rows that define some pathway that are not being observed in the literature. So it's what I call signature. It's like a combination of these rows, and you can see here that from a statistical point of view, there's going to be the problem because some of this signature overlaps, meaning that some of these rows will end up in different signature rows. So talking about the main statistical aspects of the trial, one of the key points that I hope I have covered. So the thing is, a patient is stratified depending on the big matrices of the subgroup combination. So then the outcome adaptive randomization is implemented at this level, while the treatment are monitored monthly, but not at the level of the big matrix, but on the collapse matrices, so what I call signature, and we try to update the randomization probability in such a way that when we compare a treatment, effective treatment versus the control group, we have a one-to-one comparison. And then I didn't mention that is when patients progress, so this is the outcome of interest, time to progression. When it progresses, it gets randomized one more time in this structure in order to get also some hint about the optimal sequence of treatment. So talking about the outcome adaptive randomization, so we started with fixed probabilities, randomization probabilities, and after we have enrolled 50 patients in whole the trial, then we start to change this randomization probability, and at the end of the month, we take the initial probability at the month, and then we rescale it using the probability of superiority for one treatment in one subgroup combination, so the row or the big matrix. And what is this probability of superiority? Using a Bayesian approach that I will describe soon, we take the density of the progression-free survival. For example, here are simulated data for one treatment and the control arm, and we take the difference of this distribution and we calculate the area under the curve of the difference between these two densities. And this number, like 0.8, is plugged in here squared, and the probability are rescaled. But then, as I mentioned, the evaluation of the therapies follows a similar principle, but it happens at the level of the signature. I already mentioned that the main outcome is a survival time, so I'm going to implement a Bayesian parametric model to compare the distribution of this progression-free survival time. And monthly, I will decide when to stop. These are some graduation rules that I decided based on extensive simulation studies. So basically, when I have at least 20 patients for a treatment in a signature, and I observe the probability of superiority higher than 85%, and then I put also another constraint in the big matrices in order that also those probabilities are not that low, then the treatment graduates. And on the other hand, with symmetrical number, it will stop for futility. And then there is also a maximum number of treatment. So these are simulated data just to show how the whole process it will happen. So you have one treatment compared to a control, and this is how we simulated data monthly. The two distribution will change, and the probability of superiority will reach the point where the treatment graduates after 17 months, if I remember. So those thresholds, they were decided based on extensive simulation studies, and this is because the structure of the platform study is so complex that there are no closed formula to determine how many patients or what are going to be the error. So that's why they require simulation studies that I have been implementing using R. And there's also different type of errors because the signature overlap. So you may graduate one signature that have a higher prevalence as compared to other more effective signature with a lower prevalence. So in general, these platforms are meant to be discovery trial. So what we want is to have a relatively liberal graduation criteria, and this is because then the graduating treatment will enter the validation trial. And this will assure that overall, the platform trial will give like an error rate of 5%. So here I have just shown a lot of results on the simulation studies. I will focus only on one of the scenarios that I implemented. I don't know if you can see, but generally in this setting, you have these are the true progression-free survival times for one treatment, carboplatin, that is more effective, where you have a mutation at the second biomarker, and you see that these are the general error rate, around 33% overall, and 10% for each treatment, and then you have an 80% power of graduating that treatment in that signature. And this results with more patients assigned to that treatment in that signature, and also reducing the time where the treatment stay in the trial, about 20 months against the 3 years that we fixed at the beginning of the simulation study. And this is more in detail. The Bayesian survival analysis that I'm going to implement is a parametric survival analysis, and where the time is assumed to follow a viable distribution, and we are model the gamma parameters and also the scale of this viable distribution, and we are using Monte Carlo simulation to compute the probability of superiority. So here I just presented some survival analysis with the survival package. We have more details later on, and with some Kaplan-Meier results with the simulated results, and also some presentation of survival curves, as well as results assuming Cox model or the equivalent of the parametric survival model. And I'm implementing this in STAN, since it's a Bayesian model, and I'm using the RSTAN package. Here you see the definition, if you're interested in applying yourself the model of, so you specify the data, the parameter you are interested is the effect of the covariate, and then some transformation of those parameters. You specify what is the likelihood, both for the sensor and sensor observation, and then some quantities derived from the parameter estimated. And this is just like a function to create. I don't have time to get into the details, but it's very easy to read a function to create the data set that STAN requires for the analysis, and then just with 2 lines of code running the model and getting the different parameters in terms both of accelerating failure time and proportional hazard model. And then using this model to, well, some checks are always good, and then to compare the density of the different treatment in one signature. This is the aim of the trial. Just to conclude, I just wanted to show you how did I use R in all this complex platform trial. So that has been an essential tool for me to address very different parts of this platform trial in an homogeneous way, like keeping track of the different steps that happen at the different level. First of all, using R to implement the design of the studies, and so defining the simulation and the operating characteristics of the trial, but also to actually implement the adaptive randomization that I've been showing you. And then one of the most interesting one was like I have been really using a lot of the interactive capability of the Shiny package to implement Shiny apps to explain the logical of the platform trial that is not easy to digest, and also to use dynamic reports and presentation, these slides as well. There's the link if you're interested in, and then also to develop an interactive dashboard, and here you have all the links for a prototype that I'm going to use because the people in the DSMB panel needs to keep monitoring the outcome of the trial and what's happening throughout the trial. Thank you for listening.
Thank you, Alessio. We have time for a few questions.
So the question I had was that can you go a bit more into the details of how you decided the graduation criteria that you went to? What you got?
Well, the graduation criteria are defined in such a way that you have, you don't get it wrong very often, but at the same time, when a treatment is effective, you are going to be able to graduate. It is the same question about how many people do you need and what is the alpha level you need for a test. Here, the situation is a little bit more complex. So the thing is, like, I let it vary the different parameters, assuming also data that we have on other studies to see what's going to be the effect, how much is the hazard ratio that we are going to expect, or the difference in the media survival time. And it's really trying to calibrating with this simulation scenario, which is the threshold that best give you the characteristic of the trial, basically. And try to be more flexible, because in the real life, of course, nothing is going to happen according to your simulation.